Hello friends, I am Rajeshri Patel from Svkm Institute of Sciences and Research, Gandhinagar. Today I am going to discuss about electrophoresis, and in this topic today I am going to discuss about basic principle of electrophoresis. Now let's start with the definition of electrophoresis. So electrophoresis is a technique for separation of biomolecule based on their charge, size and shape under the influence of electric field. The term electrophoresis was coined from a Greek word phoresis, which means being carried away, and the literal meaning of the word electrophoresis is to carry something with electricity. Analytically, this technique is used to study the property of single charge species or a mixture of molecule. This technique is also a preparative technique, and it is used to determine the molecular weight of proteins on one hand and DNA sequencing on another hand. Now, as I have said that electrophoresis is based on the charge of the molecule. So let's look into the details of the charge. So almost all particles like red blood cells, bacteria and many important biomolecules like nucleotides, nucleic acids, amino acids, peptides and proteins acquire either positive or negative charge upon suspension in aqueous solvent. So say for example, protein molecules have a large number of ionizable groups on their surface like amino groups and carboxyl group, and presence of these group determines the net charge density of the protein molecule, which makes it move in the electric field in a direction. But the velocity or movement of this protein molecule under the influence of electric field is depend upon the size and quantity of this net charge density onto the protein molecule. Although the net charge density is depends on the chemical groups, the number of charge particles in the molecule is modified by the nature of solvent. For example, if we dissolve protein molecule or we use acidic buffer for separation of protein molecules, then H plus ion will be more. So that's why the protein tend to become more positive, and if the basic buffer is used to separate the protein molecules, then OH minus ion will be increased in the buffer in the solution, and it will tend to become more negative. Okay, so electrophoresis is not limited to charged molecule only. We have seen that electrophoresis depends on the charged particle, but this electrophoresis is not only limited to charged molecule. Typically, uncharged biomolecules such as carbohydrates can be made to wear weak charges through derivatization as borates and sometimes as phosphate. So we can say that if we dissolve uncharged molecules like carbohydrates into the buffer, then carbohydrates may wear some weak charges. Now, even two molecules have the same charge, they might not migrate together, because if there is a difference in their molecular weight, they will have different charge to mass ratio. So electrophoresis means separation of biomolecules not only based on the charge, but it is also based on the mass of that molecule. So taken together, these differences of charge and mass, they are sufficient to ensure differential migration when the ions in the solution are subjected to the electric field. Say for example, if the molecules have less mass and carry negative charge, then maybe it can move very slowly or very fast under the influence of electric field. So this is the basis of electrophoresis. Now, as we said that in electrophoresis, separation takes place under the influence of electric field. So any sample molecules or molecular ion which migrate under the electric field, they must carry some charge. So consider a situation where a spherical particle of net charge Q is placed in an electric field. So when you placed a spherical molecule having net charge Q in the electric field, the acceleration force F will act upon the particle and this acceleration force it depends upon the net charge density of the molecule and strength of the field in which it is placed. So we can write this equation, or we can write this above relationship mathematically by equation F is equal to delta E upon D into Q, where delta E is the field strength applied and D is the distance between electrode. Q is the net charge of the particle. And now since the particle has been suspended in a solution, the particle also face a frictional force, and this frictional force occurs between the accelerating molecule and the solution. So the extent of this frictional force will depend upon the size and shape of the molecule as well as viscosity of the medium in which the molecule has been suspended. So we can write this relationship mathematically by F is equal to 6 pi r eta V, where F is the frictional force exerted onto the spherical molecule, r is the radius of this molecule, eta is the viscosity of the solution, and V is the velocity at which the molecule is migrating. So we can say that any molecules under the influence of electric field, they must face a frictional force as well as accelerating force. So we can see here there is a acceleration force in the direction of applied field, but there is a frictional force opposite to the acceleration force. So the frictional force will oppose the accelerating force generated by the electric field. So equating the force of acceleration with the frictional force, we can get delta E upon dq is equal to C pi r eta V. And if we rearrange this equation to find out the migration rate of any molecule under the influence of electric field, we get V is equal to delta E q upon 6 pi r eta d. And this equation shows that the velocity of the molecule is proportional to the field strength and charge on the molecule, but it is inversely proportional to the particle size and viscosity of the solution. So this is the basic principle behind electrophoresis.